This is the video on the photoreceptors. There are two kinds of photoreceptors, rods and cones. They are the specialized neurons in the back layer of the retina that are responsible for phototransduction, or the generation of electrical signals from light particles or photons. Rods, which get their name from their long rod-like shape, are responsible for scotopic vision or low-light vision. Rods have poor spatial resolution, but they are highly sensitive to light, making them responsible for sensitivity. Rods are, in fact, so sensitive to light that they can generate electrical signals based off of just one photon of light. Rods contain a photopigment called rhodopsin. All photopigments contain a chromophore retinol paired with an opsin, or specialized protein. Different photoreceptors contain different photopigments, which are specialized to different sections of the light spectrum. This is why different photoreceptors are responsible for generating information under different lighting conditions. Rhodopsin is a combination of a 7-transmembrane protein, or opsin, with 11 cis retinol. The opsin is arranged so it has an open compartment inside where the 11 cis retinol lives. When just one photon of light hits rhodopsin, the 11 cis retinol rotates, generating all trans retinol. This slight change is enough to launch a large cascade of events. An important thing to note is that photoreceptors operate under hyperpolarization rather than the depolarization we would typically expect from sensory neurons. At rest, a nucleotide called cGMP allows sodium and calcium to flow into the cell. When 11 cis retinol shifts to all trans retinol, an intracellular messenger, or transducin, is activated. From there, a molecule called PDE is activated, which reduces cGMP. With less cGMP, the gated sodium and calcium channels close, while potassium is still allowed to flow out. This leads to the hyperpolarization of the membrane. These events are stopped almost immediately as rhodopsin kinase phosphorylates the rhodopsin, eventually preventing its ability to signal transducin. Cones, on the other hand, are not sensitive to light, but have incredibly high spatial resolution. Cones are responsible for fine detail vision and color vision. There are three kinds of cones, each with different photopigments. They are the blue, or short, green, or medium, and red, or long, cones. They are named for the wavelengths of light they are supposed to absorb. S cones make up only 5 to 10% of our cones, while the distribution of M and L cones differ from person to person. By utilizing all three kinds of cones, we have trichromatic vision. Color blindness is due to errors in the different kinds of cone cells. All of these individuals have dichromic vision. Protanopia is from an issue with the L cone cells, while deuteranopia arises from impaired M cone cells. Both of these result in red-green color blindness. Tritanopia, however, is very rare as it impedes the S cone cells and results in blue-yellow color blindness. Though rod cells can operate in little to no light, cones are responsible for photopic vision, or vision in high lighting conditions. They actually need a high lighting environment to operate, and they are responsible for what we probably think of as seeing. Mesopic vision is when rods and cones are being used in conjunction, like at dusk or dawn, and scotopic vision, where only rods are used, occurs in the dark. This ends the video on the photoreceptors.